Hello friends, welcome to BMH Learning. In this video, we are going to discuss about continuous extraction. One of the important way of classifying extractions is based on the mode of operation, generally referred as batch or continuous. The terms batch and continuous basically points to how the sample, or you can say feed, is placed into the system, that is extraction apparatus. In this video, we would only discuss about continuous extraction. I have already uploaded video on batch extraction. You can watch that also. In continuous mode of extraction, an immiscible extracting liquid is kept flowing continuously through the solution from which a solute is to be extracted. This kind of extraction is carried out when the partition coefficient of solute is low. What does this mean? Partition coefficient here refers to the distribution ratio. In easy way, we can say the ratio of the solute between the two phases. In this type of extraction, although there is not enough time to establish equilibrium, but still a solute is extracted continuously. Now, the technique demands a special kind of extractor, and the need of extractor depends on whether extracting solvent, or we can say solvent used for extraction, is lighter or heavier than sample solution from which the solute is to be extracted. So we are going to study about the designs of continuous extraction based on the extracting solvent being heavier and lighter than the sample solution. First case, when extraction solvent is heavier than the sample solution. The extracting solvent is immiscible non-aqueous solvent and the sample is aqueous solution. The experimental setup is designed in such a way that the extracting solvent is placed in RBF, or we can say round bottom flask, and is heated. The extractor container containing extracting solvent is placed above the flask. Generally, in this container, we put half of the volume of extracting solvent used in RBF. The aqueous sample solution to be extracted is poured into the extractor to form a layer above the extracting solvent. As we know, the solvent in the RBF is being heated, so the vapor of extracting solvent gets distilled and condensed as they pass through the condenser and fall as small drops through the sample solution column. So this way, it extracts the solute and concentrate at the bottom of sample solution. So at a point when the volume of extractant rises to sufficient height, it actually overflows into the flask, or it returns to the flask. And the process is continued till the extraction is complete. The second case, when the extracting solvent is lighter than sample solution. So here, similar to the previous design, the extracting solvent is heated in the round bottom flask. The vapor of the extracting solvent gets distilled and condensed as they pass through the condenser. The droplets of extracting liquid falls into the tube as they condense. The drops pass through the sample solution and extract the solute and get accumulated on the top of the sample solution. This way, when the sufficient volume of extracting solvent is collected in the extractor, it overflows into the RBF, and the process is continued till the extraction is complete. This was all. Thanks for watching.